Welcome to Planetterrian Daily, episode 114, coming to you on July 8th, 2026. Let's get into today's research and health developments. Creatine energizes dendritic cells to strengthen an immune pathway against tumors. Scientists found that creatine strengthens one of the immune system's key cancer-fighting routes by supplying energy to dendritic cells, which then activate killer T cells. The work shows a direct metabolic link between the supplement and improved immune surveillance in preclinical models. Results remain untested in human patients. The proposed mechanism centers on creatine's role in cellular energy metabolism within antigen-presenting cells. Follow-up studies will need to test whether supplementation alters tumor outcomes in people. Now we turn to another practical metabolic intervention studied in people. People using intermittent fasting lost the same amount of weight as those following traditional calorie restriction. The fasting approach produced less sense of constant food monitoring. Researchers note this difference may improve long-term adherence for individuals who find daily tracking difficult. The study compared the two strategies directly without reporting specific participant numbers. Most people assume successful weight loss requires constant awareness of every calorie consumed. The recent comparison shows intermittent fasting achieves equivalent results while removing that ongoing mental load. Right now, as you decide when to eat rather than what to measure, your body still experiences the same net energy deficit that drives fat loss. One memorable observation from the work is that participants reported less feeling of perpetual control. That difference may matter more for staying consistent than any small metabolic edge. The practical takeaway is to test a time-restricted window that fits your schedule and track only whether weight trends downward over weeks. Simulations identified a quantum effect allowing molecular bonds to break with substantially less energy input than conventional methods require. The University of Santiago team, working with the Millennium Institute for Research in Optics, modeled how vacuum fluctuations interact with chemical structures. The approach could apply to reactions currently limited by high energy barriers. No experimental demonstration has occurred yet. Planned laboratory tests will check whether the predicted savings appear in real systems. A crystal large enough to hold in one hand exhibited clear signs of quantum entanglement. The finding demonstrates that macroscopic objects can maintain quantum behavior previously observed only at smaller scales. Researchers suggest the result may help explain properties of strange metals and support development of quantum sensors. The work provides a tangible platform for further entanglement studies. Additional experiments will explore how size and material composition influence the effect. A developed material directs thermal radiation, switches modes, and retains settings without ongoing power. The properties open routes to infrared sensors and memory devices that operate with light and heat rather than electrical charge. Researchers demonstrated control over heat direction and storage in laboratory tests. The work targets applications in energy systems and sensing. Scaling and integration into devices remain the next engineering steps. A new AI-based simulation accelerates calculations of how neutron-star mergers create heavy elements. The tool reduces computation time while preserving accuracy needed to link telescope observations with laboratory experiments. Researchers developed the model to handle the complex nuclear physics involved in rapid neutron capture. It improves predictions of explosion signatures detectable from Earth. Next steps include integrating additional observational datasets from recent mergers. While fasting shows how timing affects energy balance, a different genetic model now helps classify variants in a serious connective tissue disorder. Loss of COL5A1 in zebrafish produced cardiac defects, abnormal blood vessels, and increased hemorrhage under stress. These phenotypes mirror vascular Ehlers-Danlos syndrome features. Human wild-type COL3A1 messenger RNA partially rescued the phenotypes. That outcome indicates functional conservation despite the absence of a direct ortholog. The F0 crispant pipeline allows rapid assessment of variants of uncertain significance. Rescue with wild-type sequence provides a benchmark for judging pathogenicity. The platform can now support clinical reclassification efforts for patient variants. From rapid genetic readouts in development, we look at how sustained opportunity across childhood stages shapes later outcomes. A Boston College-led study found that cumulative educational access from early childhood through adolescence best forecast higher attainment and income for low-income youth. The analysis used longitudinal data on disadvantaged American children. Single-phase interventions showed weaker links than consistent opportunity across all periods. Results emphasize the value of sustained rather than isolated support. Policy evaluations could test scaled programs that maintain access throughout development. From human development patterns, we move to how environmental change reshapes long-term ecological interactions. Researchers tracked owl-prey interactions in Chile's Bosque Fray Jorge National Park across 2 decades of environmental change. The study, published in Ecography, measured how rising variability in temperature and resource availability affected predator-prey stability. Data showed measurable shifts in interaction strength tied to weather patterns rather than simple population changes. The semi-arid ecosystem provided a clear setting for isolating climate effects. Continued monitoring will test whether these patterns intensify with further warming. From climate effects on species interactions, we examine how migration actually moves parasites between distant sites. DNA analysis revealed limited dispersal of Diplostomum parasites across North Atlantic islands despite bird migration. The study, published in the Journal of Helminthology, sampled birds and parasites from Greenland, the Faroe Islands, and related sites. Results challenged the expectation that long-distance flyers readily move parasites between distant ecosystems. Researchers used genetic markers to trace actual transmission rather than assuming connectivity. Additional island systems could clarify whether this pattern holds more broadly. Researchers compared metabarcoding replicate detection rates with droplet digital PCR copy numbers for Atlantic cod and herring DNA in Icelandic marine cores spanning 3 millennia. The 2 methods produced consistent abundance patterns for both species. Herring DNA trends also matched reconstructed sea surface temperature variability. The study supports using PCR replicate frequency as a semi-quantitative proxy when DNA concentrations are low. Future CETA DNA work can apply this cross-validation approach to other degraded samples. A single cup of river water yielded DNA that simultaneously tracked local wildlife, pollution markers, and pathogens. The approach combines biodiversity surveys with environmental health monitoring in one workflow. Researchers showed the method works across waterways without separate sampling campaigns. It reduces logistical demands compared with traditional surveys. Expanded testing in varied river systems will assess sensitivity for rare species or low-level contaminants. A Yale study showed that targeting highly connected individuals succeeds only when the social network has particular structural features. In other topologies, influence spreads more effectively through different routes. The findings challenge assumptions underlying many public health or behavior change campaigns. Researchers modeled information flow across varied network types. Practical applications will require mapping real-world network properties before selecting targets. A Stanford study modeled how climate-driven drying and higher temperatures will interact with infrastructure and demand to increase household water bills. Some U.S. cities could see costs double by mid-century under the scenarios examined. The analysis, published in Nature Sustainability, integrates climate projections with economic and infrastructure variables for the first time at this scale. Results highlight affordability risks beyond simple supply shortages. Cities can use the framework to prioritize adaptation investments. A new report found that fewer than 1/4 of sponsorship ties provide the mutual trust, candid feedback, and active advocacy needed for advancement. Sponsorship remains critical for reaching senior roles, yet most pairings fall short of these standards. The study examined what distinguishes effective relationships from weaker ones. Organizations seeking to improve leadership pipelines can apply the identified criteria. Training programs targeting these elements could raise the success rate. Before we go, keep an eye on how the zebrafish crispont pipeline performs when researchers apply it to the first set of patient-derived Col3A1 variants submitted for reclassification. That's Planetarian Daily for today. If you enjoyed this, a rating or review on Apple Podcasts or Spotify really helps new listeners find the show. I'm Patrick in Vancouver. Thanks for listening, and I'll see you tomorrow. And if you'd rather watch than listen, find us on YouTube at NERA Network. Links in the show notes. And before you go, this show is part of the NERA Network, a family of daily podcasts on tech, science, and markets, each one a short, sharp briefing you can finish on the commute. If today earned its place in your feed, here's your next listen. SpaceX Daily, the daily companion for following SpaceX as a public company, from Starship to the stock ticker. Find every show free at neranetwork.com. This episode used AI voice synthesis of my voice. Editorial selection and analysis are my own.